All right, hey, what's going on, everybody? It's Mr. Binder here. It kind of seems like it's been a while since I did my last screencast. Uh, what you guys learned in Chapter 3 was an introduction to metabolism, and essentially what that is it lays the foundation or the groundwork for these next two chapters, cellular respiration and photosynthesis. They are complementary of each other because it's the same chemical reaction. Essentially, the products and the reactants are reversed. So cellular respiration, we see glucose and oxygen gas as the reactants in cellular respiration, and the products, carbon dioxide and water. And for photosynthesis, it's the actual reverse. We see water and carbon dioxide as the reactants, and oxygen gas and glucose as the products. Now this is important in terms of free energy, because the oxidation of glucose, essentially, that's going to take place in our cells. There's a release of free energy. All right, there's a lot of energy in between the bonds of glucose molecule, and essentially what our cells will do is break apart that glucose, harness the energy, and use it to form or reform ATP. Okay, this is very important. Okay, this is a very important process because, again, remember, ATP is the energy currency of our cells. That's what we like to call it, the currency of our cells. And essentially what ATP will be used for in our cells is to drive chemical reactions that require a specific amount of energy. Okay, again, the picture at the bottom, this describes the whole process. You guys will be reviewing this multiple times as you go through the chapter, obviously the slides. Again, I would like you guys to know this in detail. So essentially, why make ATP? ATP is crucial for cellular function. I mean, we're made up of cells. Our cells require ATP. If ATP is not present, we cannot perform the chemical reactions. Basically, our cells cannot perform the chemical reactions that are required for cellular function, meaning that we probably won't be around for quite some time. The examples I give you down here, we see ATP. There's a lot of energy in these phosphate bonds. Essentially what happens with the help of enzymes, the phosphate group will break off of an ATP and be added to a molecule that requires energy in order to perform a specific reaction. Okay, this is known as the hydrolysis of ATP. Phosphorylation is the attachment of a phosphate group to another molecule. Again, we see here the sodium-potassium pump, ATP coming into the pump, attaching. Remember, the specific enzyme that attaches the phosphate group to the pump would be your kinase enzyme. And again, once the phosphate group is attached to the pump, there's a transfer of free energy from ATP to the pump. Now the pump's able to perform its specific chemical reaction, and that is to transfer sodium ions across the gradient, okay, against the gradient from an area of low concentration to an area of high concentration. That requires energy. And this is just one example of all of the chemical reactions that take place in our cells that require ATP. Basically, the organelle that we're going to talk about is the mitochondria. The mitochondria is so important in terms of, this is the site or industrial park that's going to be making the energy. Okay, and you need to know specific structures of the mitochondria in order to understand the location of each of the different stages that take place in cellular respiration. Okay, so when looking at this mitochondria, it's a double membrane bound organelle. It consists of an outer membrane and an inner membrane. Okay, inside, it's kind of the inner portion, the most inner portion of the mitochondria here is the matrix of the mitochondria, which is specifically, it's very important for a couple of the stages of cellular respiration. And along the folds here, we see what's known as cristae. Okay, along these folds, this is where the electron transport chain will take place. Okay, along these folds here. All right, this is obviously a very, very important stage of cellular respiration because this is where we're going to see the most or the bulk portion of ATP be produced. Okay, so mitochondria is obviously very, very, very important. What we're going to talk about right now, okay, we will discuss aerobic cellular respiration and anaerobic cellular respiration. The difference between the two is if there's oxygen present in a cell. If there is oxygen present in a cell, the aerobic cellular respiration pathway will take place. If there isn't oxygen present in the cell, then there's an anaerobic pathway, and we will discuss that later. Right now, what we are going to talk about is when oxygen is present in the cell, and that's known as the aerobic cellular respiration pathway. Okay, we will take a look at anaerobic respiration afterwards. Right now, let's take a look at, hey, oxygen's in the cell, everything's ready to go. Let's take a look at the 4 stages of aerobic cellular respiration. It is divided into 4 stages. We will talk about each of the stages. Oops, I gotta cross that 4 out there. All right, glycolysis, pyruvate oxidation, citric acid cycle, the electron transport chain, okay, and it's broken up into oxidative phosphorylation and chemiosmosis. Okay, electron transport chain is broken up into oxidative phosphorylation and chemiosmosis. They're 2 processes that occur during the electron transport chain. Okay, that obviously involve some specific steps in making our ATP. Each of these stages involves the transfer of free energy. And again, the energy is coming from a glucose molecule. Well, where do we get this glucose from? Well, essentially our diet. We eat food, we break it down, we absorb it into our bloodstream, we bring that glucose into our cells, and our cells break down that glucose to make energy. Okay? Remember, when we have too much glucose, what happens in our cells, we obviously uptake that glucose and we store it as glycogen. There are obviously other processes that our cells can take in order to form glucose or other molecules that are used in ATP formation or reformation, but what we will talk about right now is essentially just a glucose molecule and the stages that occur in terms of taking the energy from a glucose and using it to reform ATP. Now, what I want you to notice here is that ATP is produced in 2 different ways. We have substrate level phosphorylation and oxidative phosphorylation. Essentially, the difference between the two, and this could be a question on your test here, okay, is that substrate level phosphorylation is the formation of ATP with the help of an enzyme. So an enzyme is actually going to be reforming ATP in this example here, or this process, substrate level phosphorylation. Okay. Oxidative phosphorylation, this process we will see, oxidative phosphorylation is used in one of the stages, okay, in the electron transport chain. And essentially what happens here is there's going to be a transfer of electrons. So here we see redox reactions. It's the transfer of these electrons that allows for the buildup of potential energy to drive a specific pump in order to reform ATP. All right, and we're going to see that in oxidative phosphorylation. Substrate level phosphorylation takes place in glycolysis and citric acid cycle, or also known as the Krebs cycle. Oxidative phosphorylation takes place in the electron transport chain. So, each stage I have written out for you: glycolysis, pyruvate oxidation, citric acid cycle, and the electron transport chain. Each stage takes place specifically inside the cell somewhere. And again, remember, the key player here, or the key organelle, would be our mitochondria, all right, that double membrane bound organelle. Glycolysis takes place in the cytosol or cytoplasm of the cell. Pyruvate oxidation takes place in the mitochondrial matrix. The citric acid cycle takes place in the mitochondrial matrix. And the electron transport chain takes place in the inner mitochondrial membrane. All right, these are great multiple choice questions. You might see them on the multiple choice questions. Okay, you will. So I'm just letting you know. Okay, I'm not hinting here. I'm just letting you know. Okay, make sure you guys understand that when I say that this could potentially be on the test, it will probably be on the test. Okay, all right, sounds good. Okay, the first stage that we're going to talk about, glycolysis. All right, this is the beginning stage of cellular respiration. Okay, can't say that this is the beginning stage of aerobic cellular respiration only because glycolysis does not require oxygen. Right? Keep in mind that as we go through these stages, we are only talking about a single glucose molecule, nothing more. A single glucose molecule, C6H12O6. That is it. Okay? So this whole process is the conversion of one glucose molecule in basically reforming the energy found in a glucose molecule as ATP. Now the first stage of cellular respiration is glycolysis, and essentially glyco meaning sugar, lysis meaning the breakdown of, so the breakdown of sugar. This is the beginning stage of cellular respiration, or the beginning stage of the breakdown of glucose. All right. This process is found in almost all organisms, both prokaryotes and eukaryotes. Prokaryotes, remember, they're different from eukaryotes because they don't contain a nucleus. We are eukaryotic, so our cells are eukaryotes, and they obviously have a nucleus, so they will also be performing glycolysis. Now, what's involved in glycolysis? Essentially, a bunch of enzyme catalyzed reactions. There's 10 of them, all right, or 10 that I want you guys to know. All right, there's 10 reactions that occur during glycolysis. Okay, each of these sequential enzyme catalyzed reactions require an enzyme. Okay, and I will be asking you guys which enzyme is used in which reaction as a matching question on the test. Okay, so there's going to be 10 enzymes you need to know or need to match with its specific reactant. Okay, and that's very, very important. All right, essentially what happens, glucose will be broken down or oxidized. Okay, it goes from a 6-carbon sugar to 2 3-carbon molecules, and these molecules are known as pyruvate. So essentially at the end of glycolysis, what are our products? 2 molecules of pyruvate. Remember, we start out with 6 carbons. We need to maintain those 6 carbons there. Okay, this is the law of conservation of mass. Okay, we see 6 carbons. We end up with 2 3-carbon molecules. We also end up with 4 ATP and 2 NADH molecules. Remember, ATP is the energy currency of the cell. NADH molecules, okay, these are co-substrates, and essentially NADH, all right, and FADH2, which we'll see later, they're important because they are essentially taking electrons from glucose. So essentially they're grabbing energy from glucose, and it will be used later to form a lot more ATP. There's 2 phases in which glycolysis, what we're going to talk about, the initial energy investment phase and the energy payoff phase. Okay. Here is the glycolysis overview. Okay, again, remember, I will be asking you guys which enzymes are going to catalyze each reaction, and there's 10 of them. So again, hexokinase obviously is going to be the enzyme that will be used to catalyze a glucose molecule. So essentially what happens here? Well, hexokinase is, well, remember the kinase enzyme here. Kinase will be adding a phosphate group to glucose. Okay? And remember those enzymes that you guys took a look at, all right? There's 6 different types. Okay? There's 6 different types. You will see some of those here in these reactions. So remember, a kinase is an enzyme that will add a phosphate group to the glucose. And we see that here. Okay? Hexo meaning 6, kinase, adding a phosphate to a 6-carbon molecule. And that would be your glucose molecule. Remember we saw isomerase? Isomerase molecules rearrange chemical groups within the cell. Well, what's different from this molecule to the next? Well, we see right here the rearranging of chemical groups in the molecule. Again, we see a kinase enzyme. This kinase enzyme is adding another phosphate group to the 6-carbon molecule. Okay, and we see aldolase here. We see isomerase. We see dehydrogenase. Remember, each of these enzymes perform a specific chemical reaction. Okay, or help to guide a specific chemical reaction. All right, remember, enzymes aren't used up in the chemical reactions. They're only there to facilitate it or help facilitate it. Okay, each of these enzymes are used to perform a specific reaction or function. Remember, aldolase, breaking apart molecules here. We see 2 3-carbon molecules now. Isomerase, again, is essentially just rearranging chemical groups within the molecule. Okay. As you go through each of these, okay, take a look at the enzyme's name. Remember, when we took a look at the 6 different enzymes that perform specific functions with as a chemical reaction is performed, okay, I want you to take a look at their names. Their names kind of give it away. Use these names, and this will help you to answer the questions that I give you in the matching section of the test. Okay. So glycolysis overview. Remember the 2 different phases, the initial energy investment phase and the energy payoff phase. What's the difference between the two? Well, in order to start glycolysis, 2 ATP need to be used here. Okay? 2 ATP need to be used. Once those ATP are used to start the whole process, the energy payoff phase, all right, is essentially 4 ATP being produced as a result. Okay? 2 NADH molecules are synthesized. Now, because we used 2 and we formed 4, the net ATP production of glycolysis is only 2. So when I say the net, even though there's 4 ATP produced, the net ATP produced is 2. All right, and essentially the 2 ATP that were produced as a result of glycolysis, or the net ATP, that will actually be used to start this whole cycle over again, the whole glycolysis cycle over again. Okay? The ATP that are produced as a result of glycolysis, again, produced from an enzyme, substrate level phosphorylation. Okay? An enzyme will actually be producing the ATP as glycolysis occurs. Or essentially what it is an enzyme grabs ADP, okay? Grabs ADP, grabs this one of the substrates, or essentially one of the reactants, okay, as a result of glycolysis, and it will take the phosphate group from that reactant and add it to ADP, forming ATP and your product. And again, you'll see that as you go back and review the picture of all the enzymes and the reactants in glycolysis. Now, essentially, that's it. We have our 2 molecules of pyruvate, we have our 2 molecules or 2 net molecules of ATP, and we have our 2 molecules of NADH. Now, this process occurs in the cytoplasm of a cell. Now, what has to happen is pyruvate needs to end up inside the mitochondrial matrix, or the matrix of the mitochondria. You'll hear both of those terms. Well, pyruvate can take 2 paths. Again, remember, glycolysis doesn't need oxygen to occur. But remember, right now, what we're going to talk about is the aerobic pathway. All right? So pyruvate can take two pathways here. The aerobic pathway or the anaerobic pathway, depending on if oxygen is present in the cell or not. If oxygen is present in the cell, pyruvate will move into the mitochondrial matrix, all right? And again, pyruvate oxidation will occur, Krebs cycle, and the electron transport chain, which is what we'll talk about. If oxygen is not present in the cell, pyruvate will actually stay out in the cytoplasm and follow an anaerobic pathway. And essentially what this is lactic acid fermentation. And we will discuss both of these processes. Right now, what we will be discussing, again, like I said, is oxygen is present, pyruvate will move into the mitochondrial matrix, okay, and we will discuss all of the other stages that occur as a result of aerobic respiration. Okay, so pyruvate oxidation. Again, this basically what happens is oxygen is present in the cell. So pyruvate will make its way through the two membranes of the mitochondria, make its way into the matrix of the mitochondria, and the next process or stage of cellular respiration, or now we will start to talk about aerobic respiration, is pyruvate oxidation. And essentially, what happens here is pyruvate oxidation essentially is the removal of a carbon group, okay, and the addition of a coenzyme A. This step is just preparing pyruvate for the Krebs cycle, okay, for the Krebs cycle. Now, pyruvate just moves across the outer membrane through large pores of the mitochondria. There's a specific protein carrier, okay, that will transport pyruvate into the matrix. Right? Once inside the matrix, pyruvate oxidation, basically what happens here is it's converted into an acetyl group. Acetyl group is a 2-carbon group. Okay, this will be your acetyl group here. Okay, acetyl group is bonded to a coenzyme A. All right. Essentially, the product of pyruvate oxidation is acetyl-CoA complex, and this molecule is going to be the starting molecule of the Krebs cycle. Right? Essentially, what happens here, there's two reactions, a decarboxylation reaction, okay? So, the removal of a carbon dioxide from the pyruvate molecule, okay, and then a dehydrogenation reaction. Essentially, the two electrons, okay, from pyruvate are transferred to form NADH. Okay, so the net reaction of pyruvate oxidation, we see 2 pyruvate, 2 NAD+, 2 CoAs or coenzyme A's. Okay, as a result, the product here is 2 acetyl coenzyme A's, 2 NADHs, okay, 2 hydrogen ions in solution, and 2 carbon dioxide molecules. Now, remember the overall reaction of cellular respiration. The product is 6 carbon dioxide. Here we see 2 of them. 2 of them form. Okay. Essentially, what pyruvate oxidation is, the acetyl group reacts with the sulfur atom of coenzyme A, and that occurs right here. Okay, that occurs right here. The coenzyme A molecule, okay, a different color here, okay, is right here. The acetyl group is right there. That came from pyruvate. This molecule here is coenzyme A. Don't worry, you don't need to know the structure of coenzyme A. Just know that the reaction occurs between the carbon or the acetyl group and the sulfur of coenzyme A. This molecule, okay, coenzyme A or acetyl-CoA, sorry, will start the Krebs cycle. Now, citric acid cycle, okay, or the Krebs cycle, discovered by Sir Hans Krebs, okay, back in the 1900s, okay, consists of 8 enzyme catalyzed reactions. Okay, so again, there's 8 enzymes that I'd like you to know for the test, and those enzymes will be basically in the form of a matching question. You need to match the enzyme with its specific reactant. Okay. The ATP that are produced in the citric acid cycle are all produced by substrate level phosphorylation. Again, the enzyme here will be reforming ATP. So, 2 molecules of pyruvate are essentially converted into acetyl-CoA. Acetyl-CoA is going to start the initial process or initial stage of the citric acid cycle. Now remember, there's 2 pyruvate molecules. So those 2 pyruvate molecules get converted into acetyl-CoA. So we have 2 acetyl-CoA molecules here as a result of essentially glycolysis and pyruvate oxidation. So this cycle occurs twice, okay? Occurs twice because there is two acetyl-CoA molecules. So we see two carbon molecule because acetyl groups, okay, two carbons, attaching to a 4 carbon molecule, producing a 6 carbon molecule, okay? Essentially, that's the beginning process of the citric acid cycle, okay? We see acetyl-CoA coming in, attaching to oxaloacetate, okay? Citrate synthase is the enzyme here that will start the actual process, okay, or the enzyme that will start the overall process here. Will take the two carbon molecule acetyl-CoA, oxaloacetate, the 4 carbon molecule here, and produce a 6 carbon molecule known as citrate, okay, and the beginning enzyme here is citrate synthase. All right, CoA, again, this is just a coenzyme. Again, they won't be in this molecule will not be involved in the whole reaction. It's just used to start the overall process of citric acid cycle. Once the process starts, CoA will release and be reformed as a result of pyruvate oxidation. All right, as you can see here, this is the overview. All right, we see acetyl-CoA. Well, essentially we see pyruvate. Okay, pyruvate will undergo pyruvate oxidation, essentially preparing pyruvate, all right, to begin the cycle of the citric acid cycle. All right, so pyruvate is converted into acetyl-CoA. Acetyl-CoA with oxaloacetate, okay, and citrate synthase, which you don't see here, is the enzyme responsible for the formation of the 6 carbon molecule citrate. Okay, this is essentially the beginning of the citric acid cycle. And this only shows one turn of the overall process. Okay, but what we can see here is the formation of 3 NADH molecules and the formation of 1 FADH2 molecule. Okay, again, we also see the formation of 1 ATP molecule. Now, because this cycle occurs twice, because there was 2 pyruvate molecules, because 2 pyruvate molecules got converted into 2 acetyl-CoA molecules, all right, and we're talking about the conversion of 1 glucose molecule. So, because there are 2 of each, this cycle needs to occur twice. So we have to multiply everything by 2. So, because there were 3 NADH molecules, there's a total of 6 NADH molecules. All right. There's a total of 2 FADH2 molecules, and there's a total of 2 ATP molecules. Now, remember, there's another product here, CO2. All right. The formation of carbon dioxide takes place as a result of the citric acid cycle. All right. There was 2 forming as a result of pyruvate oxidation. We see that there are, well, we have to multiply this by 2, so there's 4 here. All right. Now 4 plus 2 is 6. 6 CO2 is one of the products formed as a result of cellular respiration. So we see all of the carbon dioxide molecules forming through pyruvate oxidation and the citric acid cycle. Okay. Again, this is just me reiterating that all of the carbon dioxide molecules have been produced as a result of pyruvate oxidation and the citric acid cycle. Taking a look at the oxidation of 1 glucose molecule so far, we haven't even discussed the electron transport chain yet, but this is something that you are probably going to have to fill in as a test question. Okay, it will look something like this. How many NADHs are formed as a result of each of the stages? How many FADHs or 2s are formed as a result of each of the stages? Okay, CO2 and ATP. Again, I asked for the total number of net molecules. So you would say for ATP here, for glycolysis, you would say 2, not 4. Again, remember that's 2 net ATP molecules, not 4. Right? And finally, we discussed the electron transport chain. And essentially what the electron transport chain is using the NADH molecules and FADH2 molecules that were formed as a result of glycolysis, pyruvate oxidation, okay, and the Krebs cycle here. All right, they're formed, okay, so they can essentially provide the energy that's necessary to make or reform ATP. Okay, and we will discuss these kind of two processes that occur as a result of the electron transport chain: oxidative phosphorylation and chemiosmosis. Okay, oxidative phosphorylation is essentially just the transfer of electrons through a series of enzyme catalyzed reactions here. Okay, and essentially it's just the transfer of electrons through these complexes. All right, and essentially as we see the transfer of electrons from NADH and FADH2 through these complexes, we'll see, all right, we'll essentially see the movement of hydrogen ions through complexes into the intermembrane space. All right, and essentially the buildup of hydrogen ions in the intermembrane space is known as an electrochemical gradient. Okay, and this electrochemical gradient is energy, and this energy will be used to produce ATP. Remember, the electron transport chain occurs on the inner mitochondrial membrane, on those cristae, okay, that we saw earlier. And essentially, the electrons from NADH and FADH2, all right, and remember, these molecules were formed as a result of glycolysis, pyruvate oxidation, and the Krebs cycle. Okay, so as a result, okay, these molecules here will bring the electrons that were taken from a glucose molecule, all right, and bring them to the electron transport chain. Essentially, the electron transport chain is composed of 4 complexes: 1, 2, 3, and 4. We see 1, 2, 3, and 4. We also see electron shuttles right here and right here. Okay, we see ubiquinone and cytochrome C. Okay, now essentially what happens is NADH will supply electrons to complex 1. FADH2 will supply electrons to complex 2. Notice how complex 2 is a peripheral protein. It is not an integral protein. Complex 1 is an integral protein. Okay, so essentially when you see the integral proteins here, you're going to see a transfer or a movement of hydrogen ions, okay, into the intermembrane space as a result of electron transfer from NADH to complex 1. Okay, and essentially what happens here is when the electrons are dropped off to complex 1 from NADH, those electrons will move from complex to complex. Okay, complex to complex through the electron transport chain, and then finally ending up at this reaction here, forming water. All right. Okay, the driving force between the electron transport chain is known as electronegativity. Essentially, each complex has a higher electronegativity than the one before it, and essentially electronegativity is an attraction for electrons. So this complex here, complex 2, has a higher attraction for electrons than complex 1. So those electrons will be moved from complex to complex. Obviously, we see complex 4 here has a higher electronegativity than complex 3, so it wants more. It wants those electrons more. Finally, oxygen. Oxygen here is a key player because oxygen has the highest electronegativity for the electrons that are being transported through the chain. So the electrons will actually move to oxygen. Oxygen is the final electron acceptor. What happens when oxygen accepts these electrons? Well, it is converted into water, and we see water here as one of the products of cellular respiration. Okay, essentially what happens is the reaction is for half an oxygen molecule, but because we breathe in O2, okay, this obviously will not, it doesn't account for half an oxygen molecule. So what we see here is for every O2 molecule, 4 electrons are pulled through the chain, and 2 water molecules are produced as a result. Okay. Essentially, what happens in this chain of reactions? Well, again, oxygen here being the highest having the highest electronegativity value of all of these complexes, okay, electrons will be pulled through the chain. NADH is the least electronegative, but contains the most free energy, because that energy came from glucose. Oxygen has the highest electronegativity, but contains the least amount of free energy. Okay, and we can see that in the graph here. Free energy released as a result of the complexes. So NADH and FADH2 are the co-substrates here that has the highest free energy. Well, as the electrons are being passed through the complexes here, there is a decrease in free energy, okay, but higher electronegativity for each of these complexes. And then finally oxygen. All right, that free energy is essentially being either harnessed, okay, in terms of shooting hydrogen ions into the intermembrane space, or released as thermal energy. The end result of the electron transport chain is to produce this proton motive force, or this electrochemical gradient here. Okay, as these electrons are being transported through the complexes and then finally here to oxygen forming water, each complex, integral proteins here, they will be pumping hydrogen ions into the intermembrane space. Well, the hydrogen ions here will create what's known as a proton gradient. This is potential energy. Okay, and this is essentially oxidative phosphorylation right here. This is the transfer of electrons, okay, through complexes and pumping hydrogen ions into the intermembrane space. This proton gradient will be used as it's known as a proton motive force, and it will be used to drive this huge blue complex here you see, okay, known as the ATP synthase, to reform ATP. And it's the movement of these hydrogen ions through this complex here, this ATP synthase, that is known as chemiosmosis. Okay, chemiosmosis, and we will discuss that. Chemiosmosis is the ability of the cell to use the proton motive force here, okay, to reform ATP. Essentially, the hydrogen ions that were pumped into the intermembrane space as a result of the electron transport chain, or oxidative phosphorylation, these hydrogen ions will be used to move again down the gradient, or with the gradient, okay, through the ATP synthase here. As it works its way through the ATP synthase, the ATP synthase will actually reform ATP. Chemiosmosis, it relies on ATP synthase. Again, like I said, the hydrogen ions that are pushed up into the intermembrane space as a result of oxidative phosphorylation will be used, okay, to drive the ATP synthase. Okay. NADH from glycolysis. All right, essentially, NADH from glycolysis that's formed, okay, it's formed in the cytoplasm. Well, it needs to get into the mitochondrial matrix somehow. So how is NADH transported from the cytoplasm of a cell into the mitochondria? Well, there's two different shuttles here, malate aspartate shuttle and the glycerol phosphate shuttle. All right, you don't really need to know these two shuttles for the test. I just want you to understand that the NADH from glycolysis needs to be transported into the mitochondrial matrix somehow. And the two shuttle systems that NADH can take is the malate aspartate shuttle and glycerol phosphate shuttle. Essentially, it will be converted into a series of molecules until it ends up okay, inside the matrix of the mitochondria. Let's take a look at NADH and FADH2. Okay, these two molecules, okay, they're produced in glycolysis, okay, produced in pyruvate oxidation as well as the citric acid cycle. So for every NADH that is synthesized, about 3 ATP are synthesized as a result of the electron transport chain. Okay, so there's 10 total. We see 10 times 3, 30 ATP. Okay. For every FADH2 that is produced. And again, there's only 2 that were formed as a result of the Krebs cycle or citric acid cycle. There's only about 2 ATP that are synthesized as a result. So 2 FADH2 times 2 ATP, total of 4. So essentially, as a result of the electron transport chain, as a result of oxidative phosphorylation and chemiosmosis, 34 ATP are synthesized from the electron transport chain. Again, we see 2 ATP molecules produced as a result of glycolysis, 2 ATP molecules produced as a result of the Krebs cycle, so 34 plus 4. Okay, you get a grand total of about 36 to 38 ATP per glucose molecule. Right? Now let's take a look at the efficiency of cellular respiration. There's actually a question in one of your assignments right here. If there's 38 ATP that are produced in the cell, right, and the hydrolysis of ATP yields about 31 kilojoules, okay, per mole of free energy, or the release of 31 kilojoules per mole of free energy. Well, 31 times 38 is about 1,178 kilojoules per mole. Well, the oxidation of glucose, okay, essentially is the release of 2,870 kilojoules per mole. So as a result of cellular respiration, our cells were only able to harness about 41% of the total amount of free energy that comes from a glucose molecule. And what happens to the rest of the energy? The rest of the energy is lost as thermal energy, and that means heat. So I mean, again, this the efficiency of cellular respiration, 41%. It's fairly efficient. All right, it is fairly efficient in terms of harnessing the energy from a glucose molecule and using that energy to reform ATP. Cells that need a constant supply of ATP, okay, obviously our brains, yeah, obviously, right? Muscle cells, all right, these 2 cells require ATP all the time. And essentially what happens here is these 2 cells will use ATP that is formed from a different process, okay, immediately when we need that immediate rush of ATP. And this is known as the creatine phosphate pathway. Now, this is a question on your test because I will ask you, hey, is there another pathway that our cells can use when a sudden burst of ATP is required? And you will say, yes, Mr. Binder, there is. It's called the creatine phosphate pathway. And essentially what this is an immediate source of energy. Okay? So we see creatine with a phosphate molecule. ADP, creatine phosphate, okay, will come together. All right, the creatine phosphate will split, meaning that there's a lot of high energy in that bond here, okay, and ADP, okay, or ATP will reform, all right. Essentially, our cells will use this creatine phosphate pathway for a sudden rush of energy, all right. Essentially, it doesn't happen, or it's not always going to be there. When using the creatine phosphate pathway, it's essentially enough energy for about a minute, so 60 seconds. All right. Again, it's only used as a result of when our cells need a burst of ATP. Right? And essentially, what happens when we make too much ATP or when we have enough ATP? Well, even our cells will require ATP at rest. Okay? This is known as our BMR, our basal metabolic rate. Okay, but essentially what happens is enzymes can be regulated. Remember this allosteric regulation that we spoke about. All right, our enzymes can be turned on or off, essentially kind of like a light switch. Well, one of the enzymes here that will be regulated, and this will be again a test question, is phosphofructokinase. So essentially, it will be turned off when there's high levels of ATP or high levels of citrate. Again, phosphofructokinase will be turned on because there's high levels of ADP, okay, or high levels of AMP. All right. So essentially, the phosphofructokinase is regulated as a result of if there's a lot of ATP or if there isn't a lot of ATP. Okay, phosphofructokinase is the enzyme that's found in glycolysis. All right, glucose, again, when there's an excess of glucose, we know that glucose will be stored as glycogen. All right. What I'd like you to do is just review these slides, review the notes, go through the stages. You'll need to know the specific enzymes that are used in each of the steps of glycolysis in the citric acid cycle. Those questions will be asked as matching on the test. Okay. And again, please follow along with the slides. It's very important to review each of the stages, what happens specifically in each of the stages, because I would like you guys to again, write down some of the slides that I will be saying that, hey, this is going to be on the test. Okay, so please write that down. All right.